Our world is filled with an astounding variety of life forms. They come in all shapes and sizes and colors. They live in caves and coulees, on mountaintops and windswept prairie, in hidden valleys and crowded city streets. Even within the same species, we can find surprising differences. Great diversity of life is visible wherever we look. But we can also see strong similarities. The choreography of generations reminds us that differences and likenesses often go hand in hand.
We've already learned about the segregation of genes on homologous chromosomes. In this program, you'll learn about gene linkages and the general arrangement of genes on a chromosome. By the end of the program, you'll be able to explain how crossing over influences the assortment of genes on a single pair of chromosomes. So stay tuned and discover what you and Sodaria have in common.
So, there's a fungus among us, huh? Were we looking at this today?
Yes. This fungus is called Sodaria. And you know what? We share something in common with it.
What do you mean?
Oh, I thought the similarities were obvious.
What, you mean we're all in the same room?
That's not quite what I had in mind. Let's think in terms of Mendel's work on inheritance for a moment. We can use his theories to predict the outcome of cross-pollination of plants. But what if our predictions are wrong? What if something happens? What if our observations don't match our predictions?
Gregor Mendel spent 25 years in his garden studying inheritance. His work laid the foundation of modern genetics. But no one, it seems, was interested in his discoveries. His results were published, but only by the local Natural History Society. His theories went unnoticed for more than 30 years. It wasn't until the turn of the century, long after Mendel had died, that his ideas resurfaced. In the early 1900s in the United States, biologist Walter Sutton was studying the formation of gametes in grasshoppers. Half a world away in Germany, Theodor Boveri was studying similar ideas. Independently, they both came to the same conclusion, and in doing so, they put Mendel back on the genetic map. Boveri and Sutton each noticed a striking similarity between what Mendel called factors and the behavior of chromosomes. For example, Mendel's factors occurred in pairs. So did chromosomes. Mendel observed that his paired factors segregated or distributed into different gametes. So did chromosomes. Mendel's work showed that characteristics are inherited independently of one another. Sutton and Boveri observed that chromosome pairs also assort independently of each other. Based on their observations, Sutton and Boveri individually hypothesized that chromosomes carry the material of inheritance.
It's interesting how discoveries in science build on each other. Within 10 years of Sutton and Boveri's observations, the young biologist, Reginald Punnett, developed an important tool in genetics.
Punnett, as in the Punnett square.
One and the same. He developed a chart that allowed us to predict the outcome of individual matings.
No, I actually remember that.
We're going to look at one of his experiments here where he got unexpected results. He took a true breeding purple plant that produced long pollen and mated it with a true breeding red plant that produced round pollen. Now, Scott, if purple and long are dominant genes and red and round are recessive genes, what would be the genotypes of the parents?
Okay, well, you'd have capital P, capital L from one parent, and then little p, little l from the other parent, right?
Good. All right. But Sandra, what kind of gametes would these parents produce?
Well, from one parent, we'd get capital P, capital L, and from the other parent, we'd have small p, small l.
All right. Now, Scott, how many different gametes would the dominant parent produce?
They'd all be the same type, because no matter how you arrange the genes, you're still going to end up with capital P, capital L.
Okay. What about the recessive parent, Sandra?
It's also the same, just one type of gamete. Only in this case, we have small p, small l.
Good. Now, when these two plants mated, what would be the genotype of the F1 generation?
Well, according to our chart, the genotypes would be the same, but you'd have capital P, and then capital L, little l.
All right. Now, what about the phenotypes?
You'd have long purple pollen.
All right. Now, let's take this one step further now. Suppose two plants from this F1 generation were to mate. Now what kind of gametes would be produced?
Well, we'd end up with capital P, capital L, capital P, small p, capital L, and finally small p and small l.
Right, good. Now, because both parents have the same genotype, then they would both produce the same type of gametes. So let's add these gametes to our Punnett square.
Okay.
All right, so I'll be laying them down here.
That's right.
Okay. What do you think? Big E, okay.
P, L.
Big L.
Yeah.
Capital P, little L, and then it would be little P, big, right? The third one?
Yes.
Little P, big L, and then finally little P, little L. Little P, little L. Excellent.
Now, what kind of offspring can we expect from this mating?
Well, you'd have to mate them first, but you'd end up with big P, and they go up in this top square, right? When big L, big L. Is that right?
Good.
From that, we'd end up with a ratio of 9 purple long to 3 purple round to 3 red long to 1 red round, right?
Right. But what you observe and what you expect are not always the same. Here's a chart of the results of Punnett's activity based on 381 offspring. Now, here's the expected results based on the 9 to 3 to 3 to 1 ratio. What do you notice?
Well, our results don't match up with Punnett's results.
No, they don't, do they? Well, here's an opportunity to make an observation on the scientific process. How are discoveries in science made?
Probably when a predictable event doesn't turn out the way we expect.
Right. We based our expectations on Mendelian laws of genetics. Maybe we made a mistake, or maybe there's something happening that we don't know about.
So what do we do now?
I don't know. It's kind of like a puzzle with some of the pieces missing. I guess we should find out more about how genes and chromosomes work.
You're on the right track. Let's follow this up.
Although we all belong to the same species, humans are a marvelously diverse group. We owe that diversity to nearly 100,000 different genes that are located on just 23 pairs of chromosomes.
About the same time that Punnett was experimenting with sweet peas, American geneticist Thomas Morgan was conducting similar experiments on the fruit fly Drosophila. Morgan led a remarkable team of young scientists. Their work in the fly room, as their lab was called, was some of the best genetics research work of the time. Much of our understanding of crossing over comes from the studies carried out by Morgan and his students. Together Together, they discovered that body color and wing shape of fruit flies didn't assort independently. Eventually, they proposed that perhaps the genes were carried on the same chromosome. In other words, that the two characteristics were linked.
Have you solved the problem yet?
Well, does it have anything to do with the way genes behave?
You're getting warmer. Maybe these models of chromosomes will help. Remember Sutton and Boveri?
Yeah.
Both of them suggested that somehow genes might be linked. If you look at this chromosome, it has a gene for purple flower, capital P, and also the gene for long pollen, capital L. Its homologue has the small p and the small l genes.
Okay, so that means that the two genes are linked together on the same chromosome, and they would remain linked together during meiosis, right?
Correct. Now, Sandra, how many different gametes can you get from these chromosomes?
Well, on one gamete, we'd have capital P and capital L, and the other gamete would contain the genes small p and small l.
Good. So, now, if two plants that were homozygous mated, what kind of offspring would we expect in the first generation?
Well, they'd all be the same, wouldn't they? Purple long.
Good. Now, Scott, can you review for us the ratios that we'd expect if two plants from the F1 generation mated?
Sure. We ended up with a ratio of 9 purple long to 3 purple round to 3 red long to 1 red round. But our results don't match up with Punnet's experimental results.
Right. Let's take another look at this chart. But this time, we'll cover the data in the middle. Now, that will leave us 381 offspring, 296 of which are purple long, and 85 of which are red round. Now, does this help?
Well, yeah. Looks like 3/4 of the pollen would be purple long pollen, and 1/4 would be red round pollen. This ratio makes sense if the two genes were somehow linked together and passed on as one gene during gamete formation.
Well, yeah, but what about the other data? You can't just ignore it.
Yeah, you're right. We can't ignore this data over here. Let's focus on the behavior of these homologous chromosomes during meiosis. Each one will duplicate and produce chromatids. Often, chromatids will intertwine, like this. And then sometimes they'll break. And the chromosome material will be relocated from the other homologue. This is called crossing over.
So that's where we get the capital P, small l, and the small p, capital L chromosomes.
Yes.
Okay, all right, I get it now. So the purple round and the red long offspring would be the result of crossing over. The homologous chromosomes exchange genetic material.
That explains why our predictions didn't match Punnet's results. Crossing over took place, which changed our expected ratio.
Crossing over is one of the primary mechanisms to produce variation in gametes. The individual chromosomes have a combination of DNA that originates from both parents.
Did you figure out why your prediction was wrong?
Maybe we can help you solve the puzzle.
We're chromosomes.
I came from the mother, so I'm a maternal chromosome.
And I came from the father, so I'm a paternal chromosome. We both contain genes.
And as you already know, genes determine the characteristics of all organisms.
During interphase, we produce copies of ourselves. Then, during meiosis, we separate and enter different gametes. You know, chromosomes assort independently, but that's not necessarily true for genes. They often do things differently, and that's your first clue. For example, if I have the gene capital A and the gene capital B, and my homologue has the small a and small b genes, then the gametes will be capital A, capital B, capital A, small b, small a, capital B, and small a, small b gametes. Now here's another hint. What you see isn't always what you get. Chromosomes can trade genes.
It took them 8 years. Finally, Morgan had his answer, or rather 2 answers. He discovered that some genes on the same chromosome are linked together. During meiosis, when many characteristics separate independently, these linked genes stay together and are inherited as a unit. Morgan also discovered that during meiosis, some genes move from one chromosome to another by a process called crossing over. Let's go back to that sweet pea experiment for a moment. Remember, one homologous chromosome had the genes capital P and capital L. The other homolog had the genes small p and small l. It turns out that genes for color and pollen length are linked together. So the resulting gametes were capital P capital L and small p small l. The offspring of that cross were 3/4 purple long pollen and 1/4 red round pollen. But we also discovered purple round and red long pollen. How do you account for them? They were produced by crossing over. During meiosis, intertwined chromatids exchanged genetic material producing 4 gametes: capital P capital L, capital P small l, small p capital L, small p small l. When the gametes combined with each other, we observed a change in our expected ratio.
Is this the slide of the Solarian culture we were looking at earlier, Scott?
Yeah, it is. Actually, as you see, there's the ASCII there with the spores in it. Each sac has 8 spores, and they're all dark in color.
And the dark spores are from the
Wild strain.
And this one up here also has 8 spores per sac, but it's light in color because it's from the mutant strain.
Good. Now, you people made it these 2 strains. What procedure did you follow when you made it?
Well, we took some pieces of mold from the wild strain of Sedaria that produces the dark mold, and we placed them on one side of a petri dish with agar. And on the opposite side, we put the mutant strain that produces the light spores.
As you can see, they've actually started to move towards each other and have actually mated in the middle.
Oh, yeah, well done.
And then we made a slide from a sample of the middle section.
Have you checked out your results yet?
Just about to do that now.
A little more. There you go. Good, now you got it. Scott, what kind of arrangements of spores are you expecting?
Well, since they've already mated, I'm going to say 4 and 4. 4 dark, 4 light.
Here's a 4 and 4 combination right here.
But, well, look right there. We've got a 2 and 4 and 2 combination. And there's a different combination down here. How come we have so many different combinations?
And why always 8 spores per sac?
I think it's time to do a little modeling. Let's go to the table. We'll start with a review of spores. What is a spore?
An asexually reproducing cell.
Haploid or diploid?
It's a haploid.
And produced by what process?
Meiosis.
Oh, good. Okay. Suppose this represents a chromosome. Here's a centromere, and the gene for spore color is somewhere at the end over here. Now, during meiosis, these chromosomes will duplicate, line up, and form a tetrad. Now, what happens during meiosis 1?
The homologous pair separates.
Okay.
And during meiosis 2, they separate again, forming 4 separate cells. But how do we get 8 spores?
Right. And we have 4, and we need 8.
Well, how about mitosis?
That'll do it. And during mitosis, each cell duplicates again, resulting in the formation of 8 cells. And in this case, they'll all be white, light spores. So we'll have an asp with 8 light spores. If we had started with the dark gene, then we would form an ascus with 8 dark cells. Now, what I would like you people to do is to model the events that took place in mating of the moles.
Okay, well, when the 2 strains mated, each zygote would get 1 homologous chromosome from each parent. And when spores are formed, each homologous chromosome duplicates and then separates during meiosis 1. And then it would separate again during meiosis 2. That. And then finally it would duplicate during mitosis, right?
I'll get this.
Okay.
So now we have a 4x4 spore combination.
Then how do we come up with the other combinations?
Variation within the gametes. Any suggestions?
Crossing over?
That'll do it. Can you elaborate?
Well, crossing over would occur during meiosis 1. There would be an exchange in the genetic material then. And then during meiosis 2, they would separate. Thank you. And they would duplicate during mitosis. So we could end up with a 2 to 4 to 2 combination, or a 2, 2, 2, 2, as in this one.
That's well done. You people have a good idea now how you get variations inside those 4 cases.
So what would be next then?
If we can find a crossover value, we can find the distance between the centromere and the gene that produces 4 colors. So what I would like you to do is go back to the microscopes and look at 50 ASCII and record your results in a table.
Okay.
Although Alfred Sturtevant was only 19 years old, he was one of Thomas Morgan's most promising assistants in the fly room. Sturtevant hypothesized that genes are located on chromosomes, much like beads on a string. The closer any 2 genes are on the same chromosome, the less likely they will be separated during crossing over. Conversely, the further apart 2 genes are, the more likely it is they'll cross over. By measuring the frequency that genes crossed over, Sturtevant constructed maps showing the relative position of genes on the chromosomes. He assigned map units based on how often genes crossed over. For example, if 2 genes crossed over 25% of the time, he said that they were 25 map units apart. Let's take a closer look at how this works. Suppose we know that genes A, B, and C are located on the same chromosome. The frequency of crossing over is 30% for A and B, 3% for B and C, and 33% for A and C. Can you determine what order the genes are in on this chromosome? According to Sturtevant, if genes A and B cross over 30% of the time, they're 3 map units apart. B and C cross over 3% of the time, so they're 3 map units apart. We can quickly see that A and B are far apart, and B and C are close together. Now comes the tricky part. In what order do these genes occur? Is it ABC or ACB? Again, if we follow Sturtevant's work, we can figure out the answer. The distance from A to C is 33 map units. Can you figure out the right choice now? Alfred Sturtevant did his groundbreaking work on the fruit fly, Drosophila, meticulously mapping the location of their genes. This modern map shows the location of genes on one of Drosophila's 4 pairs of chromosomes. The numbers indicate distances determined from crossing over data. Today, geneticists use much more sophisticated gene mapping techniques, such as radioactive labeling and gel electrophoresis. These techniques and others are currently being used to map the human genome, that vast collection of genetic material that forms the basic human blueprint from which each of us is built. Scientists with the Human Genome Project have already located genes for Alzheimer's disease and some forms of cancer. Perhaps one day we will be able to use this information to prevent or correct many genetic disorders.
Chromosomal gene mapping has allowed us to understand the genetics of inheritance in a fantastic way. Originally, many years ago, we identified what the chromosomal makeup of our cells are. And each individual has one set of chromosomes that we inherited from our mother and one set of chromosomes that we inherited from our father. So in that particular case, we are like carrying 2 sets of Britannica encyclopedia in each of our cells. And chromosomal gene mapping has allowed us to identify exactly where on the chromosomes a particular trait is, whether it be blue eyes or whether it be the gene for insulin associated with diabetes. And in the last few years, we've been able to map many, many genes on our chromosomes. And because we can map them, we can now understand a little bit about the basic cell physiology and genetics that makes me, me, and you, you.
In 1947, American geneticist Barbara McClintock hypothesized that small fragments of genetic material could move from one position to another on the same chromosome. Though most of the scientific community dismissed her idea, McClintock persevered with her research of jumping genes. It took 20 years before the scientific community accepted her ideas, but she eventually received a Nobel Prize for her work in 1983.
Over the results.
So I see you finished your assignment. What did you get, Sandra?
Well, of the 50 ASCII we examined, only 16 showed crossing over.
So what's the crossover value then?
Well, the percentage of crossover would be 16 divided by 50 times 100. Gives us 32%.
Right. However, only half the spores are a result of crossing over. So you have to divide that number by 2. So in matings between the wild type and mutant strains of Sildaria, we can expect crossing over or the reallocation of the dark and the light gene in about 16% of the cases. Now back to the model here. Scott, what are the chances of a gene crossing over if it's located close to the centromere?
Well, not very good because the chromatids are held more tightly at the centromere. Therefore, the genes closer to the centromere would have a less chance of breaking off during the crossing over.
If the gene was located further away from the centromere?
Well, the further away it is, the greater the chance of a break between the chromosomes. Therefore, the greater the chance of a crossing over.
The richness of life on Earth is truly amazing. And although this variety may seem random and haphazard, you've learned that each organism develops from its own unique genetic blueprint. We followed briefly in the footsteps of several scientists as we've sought to explain dictions and discrepancies in our data. Mendel started us down this path with his theories about inherited factors. Punnett helped us organize our data. Morgan showed us the significance of gene linkages and crossing over. And Sturtevant explained how to map the location of genes. The story continues as new theories and new technologies combine to bring us closer to an even greater understanding of chromosomes and genes and how they work.